Okay, we're going to do the last, next to last chapter, reproduction, um, anatomy, we'll have in lab also, spermatogenesis and oogenesis, menstrual cycle, and problems. Spermatogenesis and oogenesis are going to be the trouble areas, I can tell you already. Alright, excellent website that I've got down here. It's at PBS. They have a website on the production of babies, and they do a really nice side-by-side meiosis versus mitosis comparison that you'll want to look at. It's got a text and everything on it. Okay, so here is the layout. One of the first things we're going to be starting off with is the production of sperm, spermatogenesis, and when we get the females, production of eggs in oogenesis. Look up at the top, you see reference to N. N. Okay, well, look over at about 3 o'clock where it says diploid zygote. 2N. 2N means 23 pairs of chromosomes for humans, because the chromosomes are paired. We'll talk more about that in a minute. So 2N equals 46. That means that there are 23 chromosomes that are paired. That is, one of the pair is maternal originally, one of the pair is paternal originally. They're generally talked about in pairs. So right away you see in the production of eggs and sperm, those cells are going to be haploid, not paired. They are not going to be paired anymore, the chromosomes. They will be 23 total. When fertilization occurs and the sperm fertilizes the egg, we get back to the diploid paired chromosomes. Again, half maternal, half paternal. Okay, male. We're starting out with male because he's easier. Alright. In both male and female, the primary organ is the organ that makes the sex cells. So right away, you think testes in male and ovaries in female. And every other organ is accessory. The view that you're looking at down here at the bottom left is a black and white histology slide of seminiferous tubules. You've seen them in lab already. They're all, you can see about 6 tubules in cross-section. The sperm are being made. Actually, we start out around the edges where spermatogenesis begins along the wall. And then as the different stages of meiosis occur, the sperm, as you can see, the different stages of the cells move inward from the outer wall, and we end up with sperm in the middle here, and then they're ready to move out. Okay, the view on the right, you're looking at the development of the testes in the fetus. This requires a little bit of estrogen. Remember that some estrogen is made by the adrenal glands in the fetus. The testes are not making estrogen because the testes are still immature. But if you look, right there, this tissue actually becomes both ovaries in the female and testes in the male. If there is the presence of the male Y chromosome, then the tissue, which can either develop male testes or female ovaries, will develop male. And if there is no Y chromosome with the determinating genes, it will just become female. But it's the same tissue that develops into both organs. Notice the kidneys there. Notice that this tissue is pulled downward and goes into the scrotum. And on the right you see the testis. There we go. And it's held by a ligament-like structure. It's called the gubernaculum. I've always liked that word, naculum. And you can, I guess you can barely make it out, maybe over here. And as the fetus grows in size, it's not like this pulls down. It's that the rest of the whole body gets bigger, and as the rest of the body gets bigger, the testes, which are kind of tied down here to right there at the body wall, just kind of end up at the bottom of the pelvic cavity, fall into some little folds of skin called the scrotum that you see right there. You see right there. And that's how they reside there. They're actually sitting outside of the body. We'll talk about that more in a minute. Okay. So here's a look at the testis. This is the vas deferens right there. This is the epididymis right here where the sperm are matured. But the actual production occurs right here in these lobules. It's all supercoiled tubules. You see, what's inside of here is what you see right here on a very low power. I just showed you on a higher power a minute ago. Once all these sperm are made, they all get moved over into this area, which is kind of like Grand Central Station, which is this right here. It's called the rete testis. It's kind of like the staging area where an army would land and get their troops together, get organized to go attack something. Well, this is where the sperm are getting kind of organized and regrouped. And from here they move up into the epididymis to be stored until ejaculation. There is a whitish-like membrane. In fact, you have or will remove this off the testis. This is not the same thing as a scrotum. Scrotum is going to be the skin and material on the outside you have to peel away or cut off. But it's called the tunica albuginea, which literally means coat, tunica, tunic. Albumin or albuginea refers to white, like albumin is egg white. Okay, so it's like a white coat. Now, this is a low power histology over here, but let me remind you that in between the seminiferous tubules, I'm drawing a line in between the tubules, is where the testosterone is made. There are endocrine cells in between the seminiferous tubules. They're called cells of Leydig or interstitial cells. They make testosterone. Okay, so now we're ready for spermatogenesis. By the way, the term that you see down here at the bottom, cryptorchidism, means literally hidden testes. Crypt and orc refers to testes. This is failure of the testes to descend. There can be it could be that they're half descended. They could be right above the scrotum. Sometimes one of the first things that would happen in birth is the doctor or nurse practitioner would check and make sure the testes are descended. Sometimes you can pull them into the bag, into the scrotum, sometimes not. The problem here is if they are not descended and they stay inside the body cavity, they will not make sperm efficiently because the testes are outside of the body wall, as I mentioned. It's a few degrees lower temperature out there, and that lower temperature is needed for sperm production. So in undescended testes, cryptorchidism, the male will not make a sufficient amount of sperm. Remember that testosterone is needed for for the descent of the testes. That testosterone is actually made by the adrenal glands in the fetus. Okay, so we're going to move into meiosis now. We're going to start with one of these seminiferous tubules right there. Now, there is a wall. Let me go back in. Maybe one back? Yes. Okay, so I already mentioned this. Here is the actual wall of the seminiferous tubule, and this wall has got cells lining it, and those cells are called, well, the whole wall is called the germinal epithelium, and it consists of cells called spermatogonia. One is called a spermatogonium. And these are diploid, that is 46 chromosome, cells. They have not yet made sperm. They're sitting there inactive, and they do not become active until puberty. Remember that. It's going to be very different for the female. Okay, so moving back. First, let me lay out chromosomes here. Now remember that you have 23 pair. They are in pairs. They are in homologous pairs. Here's a homologous pair. Here's a homologous pair. There's a homologous pair. Here's a homologous pair. One of the pair carries paternal, father genes, alleles, and the other of the pair carries the female. These chromosomes come together in a paired fashion at fertilization. Remember, sperm and eggs do not have paired chromosomes. They have single. Now, these pairs, what do we mean by pairs? If you look over here, it's a pretty good definition. Look. Different variations of a trait are called alleles. So here is some trait. You can see, let me erase it now. Alright, so look at this light blue over here. Same trait. Could be for eye color. Could be for the production of melanin for eye color. Now, I'm not saying it has to be the same info. It could be that this is for brown, and maybe, and let's say that came from the father, it says paternal, but from mom, there is 0 melanin for the iris, and the eye color is blue. But it's the same trait. It's melanin production for eye color. Now, down here is another totally different trait. Maybe it's for a protein for a particular chemical pathway. All right? Now, it could be that the two alleles for that trait are the same for both mother and father, or they could be different, but it's the same trait. So these are called homologous chromosomes. They code for the same traits. So you see a variety of different, I was trying to show you different ways to look at it, which might help you understand it. Now, remember that when your cells are not in mitosis or meiosis, chromosomes are single. All right? So this is a single chromosome. This is a single chromosome. It's a homologous pair, but they have not duplicated yet. Now, look down here, and this is exactly the same thing I was talking about up there. But if you look over here at the right, here is after replication. Replication of the DNA occurs in interphase. And now you see, let's say that the left comes from the dad, paternal, and here on the right is the maternal chromosome. Okay, so the dark, over here at the dark blue, you see that the paternal chromosome, the DNA has duplicated. And on the right, the maternal chromosome has duplicated. Now, you have only got 2 chromosomes here. All right? This is a chromosome here. This is a chromosome there. Now, granted, this guy right here is a duplicate of the sister right there, and this is a duplicate of the sister here, but they're duplicates. They are called sister chromatids, but they count as one chromosome. All right. Whereas here, These are individual chromosomes. That's 2 chromosomes right there. Alright, keep it in mind. Alright, so here is the layout of meiosis, and then I'll superimpose this on spermatogenesis. Alright, I'm not going to take you through the steps again of mitosis. You know it. Okay, interphase. It's the time when the cell's not reproducing. That's most of the cell life, interphase. And even though it's called resting phase, it's doubling its ribosomes, and it's doubling its centrioles, and it's replicating its DNA, and it's really doing a hell of a lot of stuff. You just don't see it doing it. Okay, now we're going to move, obviously in meiosis, the purpose of meiosis is to reduce the chromosome number by 1/2, and we're going to go from 46 to 23. There are 2 steps, meiosis 1 and meiosis 2. We have prophase, metaphase, anaphase, telophase, just like in mitosis, same phase names, in both meiosis 1 and 2. Remember that meiosis 2 is really just plain old mitosis. There is nothing different going on in meiosis 2. It's meiosis 1 that is different. Okay, so we start over here, and here, up here at the top, and here is prophase. Now remember that the chromatids, the sister chromatids, are already present in interphase. They have been duplicated. Now you see 4 chromosomes here. The big blue, the big yellow, a small blue, a small yellow. There are 4 chromosomes there. Now obviously there are actually 23 pairs. There are going to be 46 in a real cell, but we wouldn't be able to see anything, so they've just reduced it to 4 to make life easier for you. You can see that they have their sister chromatids. It looks like 8. No, there are only 4, but the 4 have duplicated DNA. Alright, now, in prophase, one difference between it and mitosis is these chromosomes, the homologous chromosomes, which you just saw on the previous thing. Those homologous chromosomes line up very close to each other. It's called synapse. The 2 homologous chromosomes sit side by side. Let me go backward here. Here, right down at the bottom, you see homologous chromosomes sitting side by side. They have their chromatids there. You see that on the picture up here. Okay, now, right over here, you have 2 chromosomes. The blue, let's say, is paternal. The pink is maternal. You see the 2 chromatids of each chromosome. They can, not always, but they will get very close. And sometimes a piece of a chromosome will interchange with another one. So a pink chromosome ends up on the blue piece, and vice versa, and we now have genetic variation. That process is called crossing over, when there is an exchange of genetic information between the homologous chromosomes, because they're lying very closely together. And you see that has happened right here. If you look at this pair right here, notice that this blue chromosome has a little piece of yellow on it, and the yellow chromosome has a little bit of blue on it, and same thing with the other set above it. Okay, so anyway, now we are in metaphase. You know that that's when the chromosomes line up along an equatorial plate. Now you're unfamiliar with this maybe because you have seen before where each chromosome is on its own and it's along an equatorial plate. Notice that the chromosomes that are homologous are still sitting side by side in that foursome. That's different than mitosis. Alright, so there's your metaphase. Now in the next one, you see that the entire chromosome gets pulled to the pole via spindle fibers and centriole. And going the opposite way, here's your other chromosome getting pulled the other way, and then you've got the 2 on the other side. Anaphase. Okay, telophase. Over here, again, the nuclear membrane begins to reform, because back here in prophase, you look over here in early prophase, nuclear membrane is still there. Later prophase, the nuclear membrane disintegrates. The chromosome becomes uncoiled, so it just looks like kind of like a mass in the cell. And you see cytokinesis, the furrowing, the pinching in of the cytoplasm to form two cells. Okay, so now we're in telophase, and we're back over into interphase. So here is interphase. I don't have a, well, yeah, I do. I have a shot of interphase right there. Two cells. Okay, so we're in interphase over here, and notice at this point in time we now have half the number of chromosomes. Look up here at the top, you had 4. Okay, 2 in a pair, 23 pairs. In that picture we have 2 pairs, 4 total. Over here in interphase, at the end of meiosis 1, we have only 2 different chromosomes. They're each with their chromatid. We haven't separated the chromatids. In a real cell, we would have only 23. This would be 23 chromosomes in each of these cells. Notice reference to N. We're not 2N anymore, we're haploid. Now remember, we still have sister chromatids. And here are the sister chromatids for the other. Alright, now the DNA is not duplicated again in meiosis 2, just in meiosis 1 at the beginning. We go into prophase, nuclear membrane disintegrates, the chromosomes become coiled and visible and blah, blah, blah. Okay, we go into metaphase now, and here is what you've seen before. Each chromosome lines up individually along the equatorial plate. But remember, they're still as sister chromatids. Anaphase. Here is where the chromatids detach, disjoin if you like. One of these chromatids is pulled that way. The other chromatid is pulled that way. We end up with 2 cells. Here are your 2 daughter cells. 2 daughter cells being made here. 2 daughter cells being made here. Remember, we started with 1 cell at the beginning. We have now a total of 4. Each of the daughter cells is haploid. Each of these cells in real life has 23. They are not sister chromatids. They are not homologous. I have some animations on this in eCampus, but you've got to think about this. Okay, now I've just taken you through meiosis, which will serve for both entry into oogenesis as well as spermatogenesis. This gets more involved, obviously. Alright, so you're looking at a seminiferous tubule right there. I've already told you that the cells begin here around the edge. These cells are called spermatogonia. They have 46 chromosomes. Now, they are not actually going to begin meiosis. They just undergo mitosis. And at the end of each spermatogonium undergoes mitosis and makes 2 primary spermatocytes. It's the primary spermatocytes that begin meiosis 1. The primary spermatocytes are 46 chromosomes. We haven't done meiosis 1 yet. They haven't reduced their chromosome number. It's the primary spermatocytes in which crossing over occurs, because it happens in meiosis 1. Good view of a high power of a few seminiferous tubules here. By the way, that's where the cells of Leydig that are making testosterone are located, right there. Alright, now, before we get going on the spermatogenesis, let me point out this picture right here. By the way, there are those interstitial cells making testosterone. So, here is a spermatogonium. These are the cells along the wall right there. And as meiosis is occurring, the cells are getting pushed inward. You can see the sperm right over here. Now, there's something else here. It's going to come up in a minute. There are some other cells here. They're called nurse cells, or sometimes called sustenticular cells. And they are taking care of the sperm. Sperm just can't do squat. They have no motility on their own yet, even though they do have some flagella. They can't feed themselves. They're like babies. They can't do anything. So these are called nurse cells because they are taking care of the sperm. They're feeding the sperm and protecting them. And actually the nurse cells also make a hormone called inhibin, which helps to regulate sperm production. You could say that they are the sperm caretakers, if you like. They're only located in the seminiferous tubules. Alright, so look over to the left at mitosis. Look at the right at meiosis. That's really what we're going to focus on. Alright, so in meiosis, again, in real life we would have 23 pairs, but again, that's way too many to have on the picture. So what you see here is 3 pairs, okay? Pair, pair, pair. They are homologous. Actually, I take that back. We have 2, 4, 6 chromosomes. They have not yet they've not yet replicated, okay? I didn't notice that they'd replicated down there. So here is a homologous pair, coding for the same traits, but it's 2 separate chromosomes. Here is a homologous pair. There is a homologous pair. Now, this is what you would see in the primary spermatocyte right there. Remember, it's 46 chromosomes total. Everything is normal. It's 2N. Okay, we start into meiosis. It's the primary spermatocyte that starts meiosis. Before we go into prophase, we're still in interphase, you can see each chromosome replicating its DNA. So the black chromosome, which you saw up here, is replicating. So now you can see 2 sister chromatids. You see 2 for the gray. You see 2 for the red, 2 for the orange, 2 for the dark blue, 2 for the light blue. Remember, we still have 6 chromosomes only, but they're with chromatids now. That happens in interphase. Now, by the way, notice that the different phases, prophase, anaphase, are not shown in this picture. It would be too big. We are now going to go into meiosis 1. Again, prophase. Metaphase. I've gone through all those steps. We get to the end of meiosis 1. Here we are at the end of telophase, going into interphase. We have 2 daughter cells. Each daughter cell has half the number of chromosomes. Chromosome 1 in black, chromosome 2 in red, chromosome 3 in dark blue. The other cell, chromosome 1 in gray, chromosome 2 in orange, chromosome 3 in light blue. Now, again, each chromosome exists as 2 sister chromatids, but that's only 3 chromosomes in each cell. You see the same thing right here. This is where we have halved the number of chromosomes at the end of meiosis 1. All right, we go into meiosis 2. Each of the 2 cells, okay, we started with 1 cell. We have 2 daughter cells. They're called daughter even though in the male. Each one now undergoes meiosis 2, which is just a plain old mitosis. Here are the 2 daughter cells of the cell on the left, 2 daughter cells of the cell on the right. We have not reduced the chromosome number anymore. I think you can still see that there are 3 chromosomes there. 3, 3, 3, except they don't have their sister chromatids. They've separated in anaphase of meiosis 2. These 4 cells from the 1 primary spermatocyte, they are called spermatids. We'll talk about that more in a minute. They are 1N. Okay, I've just taken you all through that. I mentioned the nurse cells. They make inhibin that inhibits, it regulates sperm production. Okay, so if you look at the picture over here, unfortunately, the sperm are off the screen. They are down here. You see their flagellum, but you don't see their. You might be able to just see their heads. Their heads are down here. I'll show you a better picture. So anyway, what happens is there's a maturation process right there. The cytoplasm, which is in light blue, shrinks so that the sperm, which look like that, have almost no cytoplasm. They are basically a head, which basically is just a nucleus, with 23 chromosomes. That's all. And they have a motile flagellum. And that's spermatogenesis in a nutshell. This whole process, by the way, through meiosis 1 and 2 takes over 2 months, two and a half months or so in the male. And it's happening at different stages continuously, day in, day out. Okay, so here's our sperm. Now you can really see them better. What you're looking at on the picture is spermatogenesis, which is just the maturation. Here's your spermatid. Here it is a little bit further. You can see the nucleus beginning to become predominant, and the cytoplasm is shrinking. And when you get down here, you see no cytoplasm at all. It's just a nucleus with the 23 chromosomes. And you see that flagellum enlarging, and boom, there's our boy right there. Notice on this picture below, there is a yellow thing here called an acrosome, and it contains digestive enzymes, and you might wonder why. That's because it's got to get through the wall of the egg, which is not easy to get through. So it actually digests through the wall. These guys have got a lot of mitochondria. You can see them right here for movement. Okay. I think I'll stop there. I'm not going to be going through meiosis again when we get to the female, because it's the same thing. I am going to go through genesis, because there are some differences, but it's the same meiotic phases. So this is lecture video number 1.